All right, hello everyone, and welcome back to lecture 4 for this week, going over chapter 4. Let's go ahead and jump into it. So, this lecture, we're going to be talking about the structural basis of brain plasticity. Now, at a neural level, memory is associated with changes at the synapse. The synapse is where one neuron influences another neuron. We went over this pretty early on in the course. Now, there's the finding that there are these neural correlates of memory. So, it's trying to determine how synaptic changes are correlated with memory in the mammalian brain. It's the attempt to localize the synaptic changes to specific neural pathways and to then analyze the nature of these synaptic changes themselves. It's believed that this is due to the reduced sensitivity to calcium channels, which underlines habituation. Part of this work was done by Eric Kandel and his coworkers. They measured the neurotransmitter output from a sensory neuron, and they verified that less neurotransmitter is released from a habituated neuron than from a non-habituated one. The neural basis of habituation lies in change in the presynaptic calcium channels. The underlying mechanism is reduced sensitivity of calcium channels and a consequent decrease in neurotransmitter release. Now, the neurotransmitter release in response to an action potential, it requires an influx of the calcium ions across the presynaptic membrane. So as habituation occurs, that influx of the calcium ions then decreases in response to the voltage changes associated with an action potential. Presumably, with repeated use, these voltage-activated calcium channels become less responsive to voltage changes and more resistant to the passage of calcium ions. And this habituation has been linked to a specific molecular change. So there's this sensitization response. Sensitization is an enhanced response to some stimulus. It occurs within a particular context. The organism becomes stimulus hyper-responsive to the stimulus, and there are these neural circuits that are participating in sensitization, which differ from those participating in a habituation response. And a sensitization is the opposite of habituation at the molecular and behavioral levels. What I mean by that is to really say that in response to an action potential traveling down an axon of the siphon sensory neuron, the potassium channels on that neuron are slower to open. Consequently, the potassium ions cannot repolarize the membrane as quickly as normal, so the action potential lasts longer than it usually would. The sensitization is the opposite of habituation at the molecular and behavioral levels, and there's this change that takes place in the potassium channels, whereas in habituation, the change takes place in the calcium channels. Now let's talk about long-term potentiation and long-term depression. Long-term potentiation is really important with associative learning. It entails an enduring neural change in a postsynaptic cell after an EPSP from the presynaptic cell crosses the synaptic gap. Now, the EPSP, that stands for an excitatory postsynaptic potentials. So that's what we're referring to when we're talking about that. Now, the long-term potentiation involves persistent strengthening of the synapse based on recent activity patterns. It produces a long-lasting increase in signal transmission between two neurons. In response to stimulation at a synapse, changed amplitude of an excitatory postsynaptic potential lasts for hours to days or longer. Long-term depression, or LTD, is the low-frequency stimulation produced a decrease in that EPSP, or excitatory postsynaptic potential size. If the long-term potentiation is a mechanism for creating memories. Long-term depression is a mechanism for clearing out old memories. Now, here in figure A, there are experimenters who stimulate the presynaptic neuron with a test pulse and record the excitatory postsynaptic potential at the postsynaptic neuron. And in figure B, we see that after an intense stimulation, the amplitude of the EPSP produced by the test pulse increases, and the LTP, or long-term potentiation, has taken place, where each of these dots here represents an EPSP amplitude in response to 1 week test stimulation. The discovery of LTP led to a revolution in thinking about how memories are stored. LTP and LTD form this basis for our understanding of the synaptic changes that are underlying memory. Enhanced LTP is in recruited pathways is seen when animals learn to solve problems. And the LTP produces enduring changes in the synaptic morphology resembling those seen in memory. Now, glutamate, it acts on 2 types of receptors on the postsynaptic membrane. There is the AMPA, which normally responds to glutamate, and there's the NMDA, which is doubly gated channels, and it is normally blocked by these magnesium ions. Now, that was just some background information to kind of set this part up. There are these 2 events that must occur together or rapidly for that NMDA receptor to open. There must be a depolarization of postsynaptic membrane, which then displaces the magnesium from the pore from this strong electrical stimulus. There's also been the activation by glutamate from the presynaptic neuron, a weak electrical stimulation. Strong and weak stimuli have then been paired in this instance. So in the presence of a weak electrical stimulation, because the NMDA receptor pore is blocked by a magnesium ion, the release of glutamate by a weak electrical stimulation activates only the AMPA receptor. In the presence of a strong electrical stimulation or a depolarizing EPSP, there's this strong electrical stimulation that can depolarize the postsynaptic membrane sufficiently that the magnesium ion is removed from the NMDA receptor pore. And then there's the weak electrical stimulation. So now the glutamate has been released by the weak stimulation. It can activate the NMDA receptor to allow the calcium influx, which through the second messenger increases the function or the number of AMPA receptors, or both. Now we will discuss those GABA interneurons. Experiments on the inhibitory GABA interneurons demonstrate a phenomena similar to LTP and LTD, which is ILTP and ILTD. And the I in this case stands for inhibitory. It's used to be generally believed that inhibitory neurons were not plastic, but in fact they are. The plasticity of GABAergic or inhibitory synapses plays some fundamental roles in modulating networks of excitatory neurons. And the finding of these ILTPs and ILTDs was surprising because it was generally believed that inhibitory neurons were not plastic, but we now know that they are. The plasticity of the GABAergic inhibitory synapses plays a fundamental role in modulating networks of excitatory neurons, and studying LTP mechanisms highlights neuroscientists' uncertainty over where plastic changes are located. Now, in principle, experience could change the brain in one of 2 ways, either modifying the existing circuitry or creating novel circuitry. But in actuality, the plastic brain uses both strategies. The simplest way to find synaptic change is to look for gross changes in the morphology of dendrites. Now, neurons can change their structure in response to their changing experiences the individual is having. There's the changes in the numbers of dendrites that can be used to infer synaptic changes. More dendrites then provide more connections. A new synapse can form between already connected neurons or between neurons that were not previously connected. Now, in this figure, we are seeing an image of a synapse, um, a single synapse on dendritic spine right here. And there are these three axons coming to the dendrite before the experience. And these are the three inputs to a pyramidal cell dendrite. In this case, each axon forms a synapse with a different dendritic spine that we can see here. Now, after the experience in image B, in forming multiple spine heads that we can see have kind of sprouted here, either on the original axon, can divide and innervate two spine heads, or new axons or axon collaterals, which are those like dotted outlines that we see around here, can innervate the new spine heads. And in image C, we see these single dendritic spines, which may sprout multiple synapses. Relative to the neuronal control connection that we see up top here, the number of synapses between Aplesia's sensory neuron and the motor neuron decline as a result of habituation, which we can see here. And there's that reduction. And increase is the result of sensitization, which we can see over here. Such structural changes may underlie enduring memories. So synaptic events associated with habituation and sensitization can also trigger processes in the sensory cell that can result in the loss or formation of new synapses. Researchers like Ivanko and colleagues showed that long-term potentiation in the somatosensory cortex leads to increased dendritic length and spine density in cortical pyramidal neurons. So we can see that learning, whether in simple animals such as the Aplysia or more complex animals such as rats, is consistently associated with changes in synapse numbers. The predominant view about creating new neural circuits before the mid 1990s, though I mean this is fairly recent, was that the mammalian brain does not make new neurons in adulthood. And essentially, you're born with the number of neurons that you have, and they can get lost, but you're not making new ones. However, there is evidence that neurogenesis does occur in the mammalian brain, specifically areas such as the olfactory bulb, the hippocampal formation, and possibly in the neocortex. Have shown evidence of this neurogenesis taking place, although the reason for the neurogenesis remains unclear. In adults, neurogenesis may enhance brain plasticity, particularly regarding the processes underlying learning and memory. Now, there environments in which an individual is going to be in tends to be associated with some of the plasticity. So raising rats in enriched enclosures is associated with enhanced later learning, increased brain weight, changed dendrite length, changed glial, vascular, and metabolic processes, more synapses per neuron, more astrocytes, blood capillaries, and higher mitochondrial volumes. So all of that is to say that these enriched animals performed better on many of these tasks, and the researchers then concluded that one effect of the enriched environment is that there is this enhanced later learning. Now, this is rat studies, but try to think about this in the context of humans. This ended up being applied, this type of research and thought process, and it actually influenced the foundation of the US Head Start programs, which are designed to provide healthy and enriched academic experiences for preschool aged children living in under-resourced environments. So we're seeing that so early on in life, the type of environment that you're in can actually have this lasting impact later on in life. One thing that I've heard some research about that seems really compelling is essentially if we're wanting to try to set people up to have the best opportunities in the future, a lot of that can really start with strong early childhood educational experiences. And we know that, of course, those opportunities are not equally available to everybody within the United States. So trying to find ways to fund those mechanisms, and it seems to be a huge way to inspire change and equity in the United States. Now, although the investigators did not look at other brain parts like the olfactory bulb, we can reasonably expect similar changes in different neural structures as well. And this result is exciting because it implies that experience can alter existing circuitry and it influences neurogenesis, which then, of course, leads to the new circuitry. Now, there was this other research by Chang and Greeno, and they placed patches over one eye of each rat so that the contralateral hemisphere was deprived of visual input. Then they trained the rats on a maze. They found that the visual cortex of the trained hemisphere, the one that received the input from the eye without the patch, had more extensive dendrites, which I think is interesting to see how the stimulus coming in per eye actually led to some of these changes. Now, Nudo and his colleagues, they mapped the motor cortex of monkeys, and they found striking individual differences in the typography. They had essentially monkeys retrieve food from small or large food wells. Now, the small wells, they required these dexterous movements of just one or two fingers, whereas the monkeys could put their entire hand into the larger wells. And the digit representation on the motor cortex was then larger for the animals who had to retrieve the food from the smaller wells. And then that difference in the motor cortex typography of these monkeys. The conclusion of this was then that learning new motor skills rather than simply repetitive motor use shapes the functional typography of the motor cortex. I mean, think of this as being able to like use your left arm to throw something. Like you could do it time and again, but it doesn't take a lot of skill. But if you're trying to throw like a curveball in baseball with your left hand, if you're normally right-handed, that's going to require more skill. It's going to take more effort and kind of conscious effort that's going to be going on there. And this is essentially just showing that I had mentioned where there's this large well, there's this small well, because the small well required these more fine precision movements, more effort had to take place because it was more difficult. As a result, there was a greater motor representation of the digits, the wrist, and the arm compared to that more simple task. Especially, and like we said, the digits here, because that's going to be required to actually get out the food, whereas we can see the proportion that's used for the wrist and forearm movement. Now, more recently, imaging techniques such as the transcranial magnetic stimulation and fMRI have made it post show parallel results in humans who have these special motor skills. They've demonstrated this functional organization of the motor cortex, which is altered by skilled use in humans. I'm going to change that because it just bothers me. Now, this researcher Koss showed that when the sensory nerves in one limb are severed in monkeys, the relevant part of the cortex no longer responds to limb stimulation. Deafferented cortex then begins to respond to input from other body parts. So if we think about this, for example, with like a right-handed musician who played string instruments, they could show an increase in cortical representation of the fingers on the left hand, and Braille readers have this increased cortical representation of the reading fingers. Now, if somebody was to lose a part of their body, like let's say you ended up losing your pointer finger, it's essentially like there's this real estate that's up for sale in your brain, and the cortex responds by then getting the input from other places. Those other parts of the brain move in and get mapped onto the area that used to be dedicated to your index finger. Similarly, some of this research has, oh yeah, I already talked about that as well. Now, Ramachandran, he had indirectly measured the cortical maps in individuals with limb amputations and found that when the face was stroked softly with a cotton swab, amputees reported sensations of being touched in the amputated hand. This may be used to explain some of the phantom limb pain that we've heard about. He has a book that's out. I had read at least part of it. It was very, very interesting in some of the work he's done in this area. So essentially, when we have a hand amputee's face is then stroked lightly with this cloth swab, the person then experiences the stroke as a light touch on the missing hand, as well as a touch on the face. So the deafferented cortex forms a representation of the amputated hand actually on the face because there was that real estate. The face, you're going to want to have a lot of sensory input as well. So it kind of took up and moved into that area of the brain. As the normal somatosensory homunculus, the thumb is disproportionately large. It's receiving a greater amount of information overall in this area. And we can see that shown over here as well. Now, Schiebel and colleagues found the relationship between the dendritic size in Wernicke's area and the level of education increases neuron complexity with more extensive dendritic branching out found in females. There are these large differences in trunk and finger neurons, which appeared to be in the brains of people who had a high level of finger dexterity maintained over long periods. And the general conclusion of this research was that specific experiences can produce localized changes in the synaptic organization of the brain, and that such changes form the structural basis of memory. There was also this confirmation of Schiebel's hypothesis that cell complexity is related to the computational demands required of the cell, where neurons representing the body's trunk areas have relatively less computational demands than cells representing the finger region. And cells engaged in higher cognitive functions like language, that's found in Wernicke's area, have larger computational demands than those engaged in the finger functions. Now, if we're thinking about epigenetics of memory, there are these specific sites in the DNA of neurons which are involved in specific memories, which might exist in either a methylated or non-methylated state, where fear conditioning is associated with this rapid methylation, but if methylation was blocked, there was no memory. And epigenetic mechanisms mediate the synaptic plasticity broadly, especially in learning and memory. Epigenetic changes related to experience may contribute to the inheritance of some of these phenotypes across generations. Now, the structural differences in the cortical neurons of males and female rats, it actually depends on the gonadal hormones. So the gonadal hormones can influence cell structure and behavior in human females across the menstrual cycle, but there's inconsistent data showing that. The estrogen level drops, ends up producing this increased number of dendritic spines on pyramidal cells throughout the neocortex, but then there's a decreased spine density in the hippocampus. As such, menopausal women show a decline in their verbal memory ability, and middle-aged men show a drop in their spatial memory. And we can see then the difference in some of these dendritic spines when estrogen levels are high versus low during the rat's 4-day estrus cycle, which revealed many more dendritic spines when estrogen levels are high. All right, now glucocorticoids, those are released from the adrenal cortex in times of stress. They assist in the metabolism of proteins and carbohydrates and the control of sugar levels in the blood and cells. There's steady levels of glucocorticoids that are seen with prolonged stress, which may be neurotoxic as these can kill hippocampal cells. So as of yet, little is known about the behavioral consequences of changes in the brain due to hormones. Hormones can likely alter the course of some of the plastic changes, although it may be through some of these epigenetic mechanisms. Now, there's also these neurotrophic factors in plasticity, like nerve growth factor, which stimulates neurons to grow dendrites and synapses, and in some cases, it promotes the survival of neurons. There's also the brain-derived neurotrophic factor, or BDNF. This may enhance plastic changes, such as the growth of dendrites and synapses, and it's increased levels when the animal is learning to solve problems. What about psychoactive drugs and brain plasticity? There's been shown to be some of this behavioral sensitization where the escalating behavioral response to repeated administration of a psychomotor stimulant, such as amphetamine, cocaine, or nicotine, also called this drug-induced behavioral sensitization, takes place. The sensitization is associated with increased receptors, synapses, and dendrites. These changes were localized to the regions of the prefrontal cortex and the nucleus accumbens that receive a large dopamine projection. Psychoactive drugs appear to have parallel action. They lead to increased behavioral sensitivity of their actions. So, yeah, these epigenetic changes may render the synapses less able to actually change in response to later experiences later on. So if we're thinking about some of the guiding principles of brain plasticity, behavioral change reflects brain change. All nervous systems are plastic in the same general way. Plastic changes are age specific. Prenatal events can influence brain plasticity throughout life. Plastic changes are brain region dependent. Experience dependent changes can interact, and plasticity has both pros and cons. Now, that is the end of lecture 4. We're going to go right into lecture 5 because there's not very much to that. So this is talking about the recovery from brain injury. So in this case, let's imagine we have Donna, who has a traumatic brain injury or a TBI. Although she did show some recovery, her recovery was not complete, and there were some things she couldn't do. Now, there are 3 different ways where you could recover from a brain injury. You can learn new ways to solve problems. You can reorganize the brain to do more with less, essentially trying to make the brain more efficient in some of its processing, or you can generate new neurons to produce new circuits. If we think about this like 3-legged cat solution, so if a cat loses its leg, it can usually compensate. It's not going to grow a new leg out, but instead it will walk with just the 3 legs. The same ability to compensate is also present in humans with some of the cognitive functioning. So if a person loses a certain ability, like being able to write with their right hand, they might be able to compensate for that by learning to write with their left hand. Or in some cases, if we think about somebody like Stephen Hawking, he was actually able to write and communicate by mostly using the function of his mouth. Now, the brain can form new connections and do more with less in response to injury. This is that new circuit solution. The amount of recovery is increased significantly if the person also engages in some form of intervention, like behavioral therapy, such as speech therapy or physiotherapy, as well as pharmacological therapy, like the nerve growth factor or amphetamines. So after some of those neuronal growth factor, from a stroke, it reverses the loss of the dendrites and dendrites and the dendritic spines. Then there is that lost neuron replacement solution, such as the fetal tissue implantation. This type of procedure has a limited success rate. It's usually suitable only when there's a small number of cells that are needed, like dopamine producing cells of the substantia nigra for Parkinson's disease patients. There's also the replacement of lost cells, the epidermal growth factor, where neurotrophic factor stimulates the subventricle zone, which generates cells to migrate into the striatum and eventually differentiate into neurons and glia. And the trophic factors may be useful in treating brain injury in the future. Now, it does appear that stem cells can work in this type of condition. So on the left, we have an individual who just had a cortical stroke. You can see a lot of the damage going on up here in the upper right. There was then the infusion of the epidermal growth factor into the rat's lateral ventricle. That would be over in this space, the ventricle, which induced then neurogenesis into the subventricular zone. As a result, on the right, we can see the stem cells migrated into the site of the injury and it filled in the damaged area. The cytological organization is not normal in that space because there was that introduction of the stem cells that had to produce into that area. But it's a lot better than having that gap there. That is the end of lecture 5 and the lectures for this week. Thank you all for sticking around, and I will see you again next week. Bye-bye.